In the previous lecture, I was discussing about properties of the antigens and what are the properties that govern the antigenicity. In that, I had already discussed about foreignness, molecular weight, heterogeneity of the molecule, complexity of the molecule, conformation, and digestibility of the molecule, which govern the antigenicity. Now, proceeding further, we will see some other properties that govern the antigenicity, but may not be very important. One, that is charges. Charges in terms of whether it is necessary for the antigen to be positively charged or negatively charged, or if it is neutral, can it be still an antigen? It was found that the antigens that are positively charged will induce the production of the antibody with negative charge in the antigen binding groove. The antigen binding groove is situated in FAB region of the antibody, that is fragment of antigen binding region of the antibody, where the antigen groove will have predominantly negatively charged dicarboxylic amino acids, where CO group of the amino acids will be exposed to interact with positively charged antigen. And similarly, negatively charged antigen would induce the production of positively charged antibody in the antigen group. In the sense, in the fragment of antigen binding region, in the antigen group, the amino acids will be predominantly diamino-amino acids, where their NH2 group will be exposed as NH3 to interact with negatively charged antigen. So this is how the charged antigens will interact with the antibody, and I had already discussed about the antigen-antibody interaction in my first lecture. The charged interaction is supposed to be ionic interaction, and it is stronger interaction compared to non-charged interaction. That is why if the antigens are not charged, that is, they are neither positively nor negatively charged, they are neutral, still they would induce the production of the antibody, but the antibody interaction with the antigen will be through hydrophobic interactions or through weak van der Waals forces or through hydrogen bond or through Coulomb's forces. Because of this weak interaction, the antigen can dissociate from the antibody very easily compared to the ionic interaction. The charged interaction of the antigens is taken into advantage. Depending on the charges of the antigen can be purified from the crude extract by using an immobilized antibody in a column through affinity chromatography. The antibody will interact through the charged interaction with charged antigen, and with the result, antigen will be bound to the antibody. Then the column can be given a thorough wash, and this bonding of the antigen antibody will be very firm because it is an ionic interaction. After thorough wash, all the other unnecessary materials can be washed off. Then a suitable buffer with a different pH can be added to the column, which releases the antigen in the pure form without any contamination. And such purely extracted antigens are generally used for immunization purpose, that is for the vaccination, or to develop antibodies in animals to prepare diagnostic kits. So the charges on the antigens play an important role as far as interaction with the antibody is concerned, but it is not absolutely necessary. Coming to the next property, that is accessibility of the antigenic determinants or antigenic epitopes or immunodominant groups of the antigen. We had discussed earlier the amino acids like phenylalanine, tyrosine, glutamic acid play a major role in determining the strength of the antigenicity with respect to the antigenic domains. They play a major role in proving an antigen to be strong or weak. Presence of these amino acid residues, that is tyrosine, glutamic acid and phenylalanine in a given antigen, makes it very strong antigen. Based on that principle, a model was prepared by Sella et al. In 1966 to explain the importance of the strong antigenic domains as well as the conformation or structure of the antigen molecule and the orientation of these antigenic domains. They created artificially the molecule which is a heteropolymer in terms of using a polylysine polypeptide chain as a backbone, as shown in the diagram, the extreme left diagram. The backbone is polylysine chain to which the side chain linkage of polyalanine was attached. So on one side 3 polyalanine chains were attached, on the other side 2 polyalanine chains were attached. By attaching polyalanine by a peptide bond to polylysine chain, this created the whiskers on the backbone of the polylysine. The polyalanine side chain linkage is called as whiskers. Even though this molecule is heteropolymer made up of 2 different amino acids with large molecular weight, when it was introduced into the rabbits, it did not induce the production of antibody. So therefore, what they did to the same molecule, they modified a little bit by introducing in the side chain linkage the immunodominant groups like tyrosine and glutamic acid. The dark circles represent the glutamic acid and the light circles represent tyrosine. By introducing these side chain linkages on the whiskers polyalanine side chain in various capacity, some side chain linkage had 2 glutamic acid residues and 1 tyrosine, and some had 1 glutamic acid residue and 2 tyrosine residues, like that. It varied, but it made a difference. And then when this molecule was introduced into the rabbit or injected into the rabbit, the molecule produced the antibody. It induced the production of antibody. So that indicates that the immunodominant groups like tyrosine and glutamic acid presence in the side chain linkage exposed exteriorly on the polyalanine chain plays a role in making this molecule antigenic. Otherwise, in the absence of these 2 residues, that is tyrosine and glutamic acid, the molecule, that is polylysine polyalanine heteropolymer, was not antigenic. It was not capable of inducing the antibody production. They went further ahead by modifying the molecule as shown in the middle diagram. In the middle diagram, they maintained the backbone of the molecule as same polylysine, that is in the middle, and to that they attached the whiskers by reversing their attachment. Refer to the extreme left diagram where the tyrosine and glutamic acid residues attached to the polyalanine chain are exposed outside. Now, in the second molecule, the alanine chain is reversed in such a way that the tyrosine and glutamic acid residues got attached to the polylysine polypeptide chain backbone, and the polyalanine chain attached to them was exposed outside. That was the only change that was made. There was no change in the molecular weight. There was no change in the types of amino acid residues. It remained the same as the first molecule, but the conformation was changed. Here the structure is changed. The attachment of polyalanine chain through the tyrosine and glutamic acid residues to the polylysine chain was haphazard. Even though in the diagram it is shown very symmetrical, but it was not symmetrical. It was haphazard. It could attach anywhere it pleases, but through peptide bond with the polylysine chain. That second molecule with a different conformation or structural property, but having the same molecular weight as the first molecule, was introduced into the rabbit, was given as an injection, and they found that the second molecule failed to induce the production of antibody. Then they continued with the third model where they did some more modification. The backbone now is made up of polylysine polyalanine polymer, where there is alternating units of polylysine and polyalanine peptide. Say for example, 10 amino acid residues of polylysine peptide bonded with 10 amino acid residues of polyalanine, followed by once again polylysine and polyalanine. Like that, they prepared a backbone with the alternating unit of polylysine and polyalanine peptide, and that backbone, to which the whiskers were attached in the similar manner as it was attached in the second molecule, with tyrosine and glutamic acid residues turned inwards, and the polyalanine chain was exposed outside. Here, the difference was the polyalanine chain could attach, that is, with the glutamic acid and tyrosine residues, could attach to the backbone only at polylysine residue site. It could form the bond at polylysine residue site. Therefore, the whiskers were attached almost in the symmetrical manner compared to the second molecule, even though the tyrosine and glutamic acid residues were turned inward. But the molecule was more symmetrical compared to the second one. This molecule now was introduced into the rabbit. It was given as an injection, and after some time they found that this molecule was capable of inducing the antibody production. So this clearly indicates that it is just not the complexity and only conformation. Of course, the conformation plays a major role. The exposure of or accessibility of the antigenic domains is very, very important. The third molecule, even though the tyrosine and glutamic acid residues, being immunodominant groups, were turned inward, the conformation of the molecule, three-dimensional structure of the molecule, was capable of exposing these antigenic domains to the antigen presenting cells to recognize them as antigenic epitopes, and with the result, the third molecule succeeded similar to the first molecule in inducing the antibody production, whereas the second molecule, which did not have a symmetry, proper symmetry, of course the conformation was three-dimensional, probably it was a globular protein, but the inward turn of the tyrosine and glutamic acid residues was not exposing these antigenic determinants to the antigenic presenting cells. So probably that is the reason that the middle molecule failed to induce the antibody production. So therefore, from this experiment, it can be concluded that all the other properties that we had discussed earlier, that is molecular weight, conformation, heterogeneity, then digestibility, can be answered from these artificially constructed molecules by Sella, which explains it is just not the only presence of the antigenic domains or antigenic epitopes. They need to be exposed, they should be accessible. And accessibility of the antigenic epitopes play a major role in induction of antibody production. So let us move about what are the other factors that govern antigenicity or play a major role in proving a molecule to be antigenic. Genetic constitution is one of the important factors. An antigen could be a very potent antigen with strong antigenic determinants, might have successfully induced the production of antibodies in animals or some other individuals, but sometimes it may fail to induce the production of antibody in some individuals. The reason being the genetic constitution of the individual plays a major role in recognizing a particular antigen molecule to induce the production of antibody or immunity. As far as human individuals are concerned, each one is unique in genetic constitution except the identical twins, whose genetic constitution will be almost the same. Whereas in a family of brother, sisters, father, mother, or sister, sister, brother, brother, each one of them will have a different immune response against an antigen because of their genetic constitution. There is variability in immune response in each individual. You can take an example of coronavirus infection itself, which had happened 2 years back. You know that there were cases where the reaction was very, very severe in some individuals after the infection. And those individuals, despite the medical assistance and medical interference, did not survive. At the same time, there were individuals who were infected, showed moderate reaction, and responded to the medical assistance and recovered back. There were also cases where the severity of the COVID infection was very, very mild in some people, and they recovered very fast from the infection. So from these cases, it is very clear that in the human population, genetic variation and genetic constitution plays a major role in determining whether an antigen molecule is going to induce immunity in a person or not. Sometimes it may cause severe reaction, which may be detrimental, and it will be overwhelming reaction and may lead to death of the individual. That is purely because of genetic constitution. So coming to the second point, that is about the dosage of antigen. Whether a particular dose of antigen is essential to induce the immune response. Yes, it is very much essential, and the dosage of the antigen is calculated based on the body weight of the person. And the chart is readily prepared by CDC, the Center for Drug Research, and this center has already prepared a chart where the individuals of different body weight, how much of antigen in the form of vaccine to be given. Just to give an example, there are individuals who are ranging in the body weight between 45 to 60. Some individuals have a body weight range from 60 to 80, and some have a body weight ranging from 80 to 120 also. The dose that is recommended for a person who is weighing 80 to 120, if the same dose is administered to an individual who is having a body weight in the range of 40 to 60, it will be excessive dose. And this excessive dose administration of antigen into an individual will make the person totally immunologically paralyzed as far as that antigen is concerned. In the sense, there will be overdose of antigen in his body. Excessive antigen will be circulating in his body for several days, which results in saturation of almost all antigenic receptors in the immune cells. And those immune cells, because of the saturation, oversaturation of the receptors in them, would start developing tolerance to that antigen. When almost all receptors of the immune cells are going to be engaged by large quantity of antigen, those immune cells which are supposed to induce the production of the antibody will develop tolerance against that antigen because of the overwhelming response of the antigen. With the result, the person will develop tolerance and will not respond to that antigen in future. In the sense, will not be able to produce the antibody or immune response against that antigen in future. It is permanently the person will have developed tolerance. The antigen will behave as though the self antigens, in the sense we have in our body the self antigens in large numbers, and because of the large number of self antigens, immune cells will develop tolerance to these self antigens. They will never develop immune response against the self antigens because there are large number of these self antigens coming in contact with antigenic receptors, and the immune cells will never get stimulated. It would respond in the same way for the large amount of antigen injected into a person as a vaccine. So therefore, it is dangerous for the person who has been given excessive dose of the antigen of a particular pathogenic bacteria or a virus, would not develop immunity against that pathogen. Later, in the real life situation, if the pathogen infects him, the person may succumb to the infection because immune response will not be there and the pathogen is deadly, which would produce toxins, or it could cause some damage in the system of an individual, resulting in death of the individual. Therefore, it is very important to note that dose of an antigen is very important, which is based on the body weight of the individual, and it need to be administered strictly as per the recommendation of the dose. Similarly, when the antigen is administered in a very low dose compared to the body weight, the low dose antigen may get lost in the body, which will not be recognized by the immune cells, because according to the proportion of the body weight, if the antigen was administered, probably it would have been recognized by the immune cells. But when it is given at a very low dose, it may get lost from the body excreted from the body, may not be even processed by the antigen presenting cells. With the result, it may also cause danger to the individual if such a vaccination is done where the dose recommended was not given and a very low dose was administered, and thinking that the person is immunized against a particular pathogen through vaccination, the person remains complacent, but when the real life situation, when the pathogen infects him, there is a possibility that he or she was not immunized because of the low dose administration. So therefore, one need to take care that as far as antigen or vaccination is concerned, it should not be of low dose nor it should not be of very high dose. Both are detrimental. It should be proportionate to the body weight as recommended by CDC. Now coming to the routes of administration of antigen, that is routes of vaccination. It is also very important that one need to follow the schedule strictly as far as routes are concerned. If the vaccination is to be given subcutaneous, that is just below the upper layer of the skin, subcutaneous, the angle of the needle for this injection will be less than 45 degrees. One can see after the injection the appearance of a bubble-like situation below the skin because it is very superficial injection. So subcutaneous injection, when it is recommended, it needs to be given subcutaneous only and it cannot be given intradermal or intramuscular, because it is necessary for that antigen to be administered in that region for its proper recognition and distribution into the body to induce the immune response. Now, which vaccination is done or antigen administration is done subcutaneously? It is observed that MMR vaccination and meningococcal vaccination are done subcutaneously. Then intradermal, that is slightly deeper into the skin layer, that is the dermal layer of the skin, where the angle of the needle will be somewhere about 60 degrees. It is deeper layer of the skin. Generally, rabies vaccine is administered intradermally. And what are the vaccines that are administered through the intramuscular route? It is observed that BCG, DTP. DTP is diphtheria, tetanus, pertussis vaccine. Hepatitis A and B vaccine. Human papilloma virus vaccine, that is HPV vaccine, are administered through intramuscular route. There are also vaccines that are administered through the oral route because these pathogens infect the individual predominantly through the oral route through gastrointestinal tract. Such vaccinations are recommended as per oral administration. Now, what are the vaccines that are administered per oral? Are rotavirus, cholera, typhoid, and polio vaccine. Polio vaccine in short form is called as OPV, oral polio vaccine. You know, rotavirus, cholera and typhoid infect a person through the GI tract. Polio, which causes the damage to the nervous system, but its main route of infection is through the GI tract. It colonizes the intestinal crypt, the virus, and then spreads the infection through that crypt area. Therefore, polio vaccine is also administered through the oral route. The intranasal vaccine is a recent development where the vaccine is sprayed into the nasal passage and the vaccine gets absorbed through the mucosal lining of the nasal passage. first vaccine to have been developed is for the influenza virus. The influenza vaccine was administered through intranasal route and it was found to be successful. Now recently, Bharat Biotech has developed the corona vaccine against the coronavirus that could be given through the intranasal route, and it has been found to be successful. So, routes of administration for immunization, that is for vaccination, need to be strictly followed as recommended for particular vaccines, and the routes cannot be changed. Now coming to the another important aspect of an antigen. There are some antigens which by themselves cannot induce the production of antibody, but they are antigenic, and such antigens which by themselves cannot induce the production of antibody were given the name Haptens or Haptens. As shown in the slide, the spelling is Haptens, Haptens or Haptens. This term was coined by Landsteiner, who discovered the blood group. He observed that there are certain antigens which by themselves cannot induce the production of antibody, but on combination with some other molecule which is antigenic, they will become antigenic. To give certain examples, certain drug molecules which are organic in nature, which are administered to the patients for certain types of diseases, when they enter the body, after their circulation in the blood, sometimes some drug molecules may form complex with your own body protein. And the drug molecule per se is not antigenic, but on combination with a known protein in your body, it may behave like antigenic, as well as it could induce the antibody production against its own epitopes as well as the protein with which it associated. Let us take the example as represented in the slide. The hapten here that is considered, that is partial antigen that is considered, is DNP. DNP is nothing but dinitrophenol. Dinitrophenol is a simple organic molecule, a phenolic molecule with nitro group. The dinitrophenol, you can compare that with a drug molecule. When it is injected into a rabbit or rat or mice, it will not induce the production of antibody on its own. So therefore, in the right hand side it is shown antibodies none. Same time, if you take another example, that is a protein carrier, that is bovine serum albumin. A bovine serum albumin is a globular protein. When it is introduced into a rabbit or rat, mice, if it is injected, it can induce the production of antibodies against it, which is called as anti-BSA. So the bovine serum albumin is an antigenic molecule, strongly antigenic molecule. Now if you combine, in the sense you conjugate dinitrophenol in the form of bonds with bovine serum albumin at different levels, in different intervals, you bond it. DNP is bonded to bovine serum albumin. Therefore, BSA becomes the carrier of DNP. This now you can compare to a drug molecule in your body binding to a known protein in your body. So now you get hapten protein carrier. The hapten is DNP is attached to the protein carrier, that is BSA, and when it is injected into the rabbits surprisingly will produce 3 different types of antibodies. One antibody will be recognizing only DNP, that is dinitrophenol, as the antigenic epitope. The another antibody that will be produced will be against the amino acids of the bovine serum albumin. The peptide chain will have several different amino acids, and probably there are immunodominant amino acids like tyrosine, phenylalanine, glutamic acid in the bovine serum albumin. Those will be recognized and antibodies will be produced against those antigenic domains, that is of the BSA. Then the third antibody will be against the DNP BSA overlap region. Wherever the DNP, that is dinitrophenol, was bonded to the bovine serum albumin, in that region, the overlap region of the bovine serum albumin and DNP will show the antibodies. The antibodies will recognize the overlap region. So therefore, three different antibodies are produced. Now from this example, it is clear that a known antigenic molecule which acts as a carrier to a hapten, which is a partial antigen which by itself is not antigenic and cannot induce the antibody production, will now start producing the antibodies because it has combined with a carrier molecule which is antigenic. Let us see in the diagrammatic representation for the previous explanation. As I had mentioned, the bovine serum albumin is a globular protein which is shown here with the blue globule, to which the hapten molecule in the form of triangles, red triangles or pink triangles, is attached. And this hapten molecule, you can take it as an example, DNP. The bovine serum albumin also has its own antigenic epitopes which are shown in two different forms, which are strong antigenic epitopes, and the hapten molecule is bound to the globular protein. This molecule now, when it is introduced into the rabbit, as I had mentioned earlier, you will see three different types of antibodies. See the first antibody at the top. That antibody will recognize only the DNP, that is the hapten. It will bind only to the hapten. It can recognize the hapten, which otherwise, if DNP only was injected into a rabbit, it would not have been able to produce the antibody. Now, you have made a hapten molecule to be recognized by an antibody. Second molecule, that is in the middle, you see, the antibodies are produced only against the antigenic epitopes of the protein carrier, that is BSA antigenic epitopes. And here there are two different epitopes and two different antibodies against two different epitopes. As I had shown here in the diagram, there is two different epitopes and two different antibodies against those two different epitopes of the carrier protein, that is BSA. The third antibody that is at the bottom, you will see that it will recognize both, that is the DNP as well as the bovine serum albumin epitope at the overlapping region, and this antibody will combine simultaneously with DNP as well as bovine serum albumin antigenic epitopes. So from this one can say that hapten molecules, which are partial antigens, which could be drugs, could lead to such complications in the human body if they induce the antibody production, and one would also result in production of antibody against one's own protein, which becomes the carrier of the drug molecule, and that would result in certain detrimental effects in a person, where the antibodies, when they bind to the known protein of an individual, which will be important for that person for certain functions, and that protein will get destroyed by the antibody after it binds to it, because the hapten made that protein to carry it and made that hapten protein carrier molecule to be antigenic. So these complications will result in some detrimental effects in the human beings. Therefore, sometimes the drugs need to be withdrawn and new drugs need to be administered for the treatment of certain diseases. So this is what happens with the partial antigens. Let us move ahead further with some other examples which were explained by Landsteiner, where the antigenic epitopes in terms of the hapten as well as the antigenic determinants in the hapten, how they are important in determining the antigenicity and antigen antibody reaction, which is highly specific with specificity. He took an example of aniline. Aniline is a benzo containing or benzene ring containing with amino group substituted molecule, which is called as aminobenzene. So aminobenzene, which is aniline, having an NH2 group, when it is substituted with furthermore group like COH in the ortho position with reference to NH2 group, which is called as orthoaminobenzoic acid. When the COH group was substituted in the meta position, it is called as meta-aminobenzoic acid. And when the COH group is substituted in the para position, it is called as para-aminobenzoic acid. Now, from the original parent compound, that is aminobenzene, three other compounds were produced, three other derivatives were obtained, which are of different structure, in the sense one is of ortho substitution, another is of meta substitution, another is of para substitution of COH group, in addition to the NH2 group which is already there in the parent compound, that is aniline. When these four different molecules of the aniline derivatives and aniline are used to conjugate with a polypeptide, either it could be a bovine serum albumin or it could be polylysine. Polylysine to be on the safer side, why? Because polylysine by itself is not antigenic. We have proved that being a homopolymer, it cannot be antigenic. Now, when you make these like aniline to react and bond with polylysine at different regions, it will bond with polylysine and it will be forming hapten protein carrier. Now protein here I refer to polylysine, which is the polypeptide. Similarly, orthoaminobenzoic acid will be conjugated or bonded to the polylysine chain. Similarly, meta-aminobenzoic acid will be conjugated with another polylysine chain and para-aminobenzoic acid with the another polylysine chain. Now you have four different molecules combined with the polylysine chain having four different peptides. Now these four molecules are injected separately into four different rabbits, and all four molecules were successful to induce the production of antibody. The antibody in terms of antiserum is represented in the extreme left hand corner of the table. So antibody against the aminobenzene recognizes aminobenzene only, and it is shown by 3 pluses indicating it is a good strong reaction, and it does not recognize the other antigens, that is other hapten protein complexes like orthoaminobenzoic acid, meta-aminobenzoic acid and para-aminobenzoic acid. Therefore it is shown as 0. So the antibody against the aminobenzene recognizes specifically only aminobenzene. Similarly, the antibody against orthoaminobenzoic acid recognizes only orthoaminobenzoic acid, not any other peptides. Not any other peptides. So this is the reason that specificity can be proved from this example, where the antibody is produced against the four different molecules which were formed as a complex as hapten with the polylysine, are capable of specifically recognizing their own antigens or the haptens and not other haptens. This proves the specificity. Now here the specificity lies with NH2 group, and the NH2 group of the molecule is common in the aniline as well as the derivatives, it is in the same position, as well as the specificity lies with the COH group, which is in the ortho and meta and para position COH group. So accordingly, the antibodies recognize these positions of the antigenic domains NH2 and COH, and these antibodies showed highly specific reaction with reference to these different haptens with reference to those antigenic determinants. And it is observed that the meta-substituted hapten, meta-aminobenzoic acid, was found to show much more stronger reaction followed by para-aminobenzoic acid compared to the aminobenzoic acid, orthoaminobenzoic acid, or aminobenzene. So the positions of the antigenic determinants determine the specificity, and these antigenic determinants are highly specific. It could be even SH group which can act as an antigenic determinant, and they are very strong determinants. Therefore, here you don't see any cross reactivity of the antibodies with other antigens or cross reactivity of the antibodies with other haptens. Now let us move further ahead with slight modification. Landsteiner took aniline, same molecule, but here now the substitution was with different other molecules, like one was substituted with the chlorine group, that is para-chloroaminobenzene, that is the second molecule. The third molecule was substituted with a methyl group, which is para-methyl aminobenzene, which is also called as toluidine. And the fourth molecule was nitrogen oxide, and that is para-nitroaminobenzene. All were having para substitution, but NH2 group was in the same position as far as the parent molecule is concerned in other haptens. Now these four molecules were once again made to combine with polylysine polypeptide separately, and they bonded with polylysine polypeptide at different regions, and after construction of these different molecules, each one of them were injected into the rabbit, and they waited for the antibody production. Surprisingly, when the antibodies were produced, we will see that the antibody produced against the aminobenzene will not only recognize aminobenzene, was also capable of reacting with para-chloroaminobenzene, was also capable of reacting with para-methyl aminobenzene and para-nitroaminobenzene haptens combined protein. You can see that none of the columns having 0. There is some reaction that is showing cross reactivity. Take the second example. The antibody produced against the para-chloroaminobenzene showed the same intensity of the reaction as aminobenzene. You can see both of them are showing plus, 3 pluses. And it not only recognized the aminobenzene, it also recognized the para-chloroaminobenzene, and there was reaction with para-methyl aminobenzene and para-nitroaminobenzene. So you can see in this table that there is no place where the 0 is represented. Almost all molecules which showed cross reactivity with the antibodies of different one. There was no specificity as far as the antibody is concerned here due to cross reactivity. From this example, it is very clear. Now if we take the previous slide and see here, only common feature that we see here is the NH2 group, which is present in the same position in all the 4 molecules. And the NH2 group, as I had mentioned earlier, is a strong antigenic determinant. And this proves that the molecules, that is the groups like chloride, methyl, or nitrogen oxide or nitrate groups are not capable of determining antigenicity. and they being there, they were not able to determine the antigenicity. But the NH2 group itself determined the antigenicity. That's why in the antibody response, there is cross reactivity with respect to NH2 group only. It is not recognizing the chloride or methyl group or nitro group. Hence, it is very important that antigenic determinants like the NH2 and COH groups as well as SH groups, if they are present, play a major role in determining the antigenicity. And from this Hapten example, which was conjugated with the protein carrier, Landsteiner could explain the specificity of antigen-antibody reaction and what determines the specificity of antigen-antibody reaction. It is very clear from this example that the major domains of the antigens such as NH2, COH, SH, all these groups determine the antigenic epitopes. Therefore, such antigens having such groups will have highly specific antibodies, whereas antigens having very weak antigenic determinants like chlorides, CH3 and NO2 groups will show cross reactivity as represented in this table. So therefore, sometimes when there is a pathogenic infection, the antigenic determinants of the pathogen determine whether there is specificity or cross reactivity. It is observed that in some of the autoimmune responses, the antibodies produced against certain pathogens, their antigenic determinants cross react with the antigenic determinants of the self proteins of the human body, resulting in induction of autoimmune response, which will be discussed separately in autoimmunity. So cross reactivity as far as antigen antibody reaction is concerned is highly dangerous, because the antibody produced against the pathogen, if it starts acting against one's own body proteins and antigenic determinants, it will be detrimental for the person. But it is a rare incidences and strange incidences it would happen, but it is not very common and not general. Therefore, one is saved from the cross reactivity of the antigen antibody reaction. Most of the time it is highly specific, just like the way the enzyme substrate reaction takes place. So this is as far as the antigens, their properties, and what are the factors that govern antigenicity, and what are partial antigens like haptens, and why they are dangerous if they cross react or they react with one's own body protein has been explained, and will stop here. Thank you very much.